Assalamualaikum and good morning. Welcome to Biologi Profesor Lectures Pusat Asasi UiTM Dengkil for this semester. My name is Mazhani Muhammad and it's a pleasure to be the moderator for today's lecture. First and foremost, I would like to welcome our guest professor yang berbahagia, Prof. Dr. Muhammad Faiz Fong bin Abdullah from the Faculty of Applied Science UiTM Shah Alam. And welcome to all biology lecturers and students. Before we begin our lecture, let us all recite Al-Fatiha. Okay, so on behalf Pusat Asasi UiTM, I would like to extend a very warm welcome to Prof. Dr. Muhammad Faiz. So before we start, let me introduce a little bit of his education background. So Prof. Dr. Muhammad Faiz is a lecturer in the Faculty of Applied Science, UiTM Shah Alam. He graduated with a degree in microbiology from University of Malaya and master degree in biotechnology and molecular biology from University of Western Australia. He completed his PhD in biochemistry from University of Oxford. His main field scientific interests are molecular biology, microbiology, biosafety and biosecurity. So in our lecture today, we are going to discuss chapter 5 on cellular respiration. So to all students, if you have any question, you can write your question in the chat box. Okay, so without further ado, I would like to invite Prof. Faiz to begin his lecture. Over to you, Prof.
Okay, Assalamualaikum, salam sejahtera and good morning everyone. So thank you Dr. Mazani for the nice introduction. Okay, I just confirm that you can hear me clearly. So I assume that everyone can hear me clearly. Right, okay, thank you. Okay, so it's good to see you students again. I'm not sure I have spoken to you before last semester. Okay, but it's always good to be among young, eager minds, okay, ready to learn the latest in biology. Okay, so once again, thank you for inviting me to deliver this lecture. Okay, so this semester I thought I would challenge myself a bit and pick a title at random, and I end up with a cellular respiration. Okay, I used to sort of dislike this chapter during my student days because there's a lot of things to memorize, all the pathway and all the equations and so on. But technically, I would say this is one of the most interesting chapter in biochemistry. Okay? So cellular respiration is one of the most important process in biology. Okay? You can don't eat for a few days, you can don't drink for a day and so, but you can't stop breathing and respiring for more than 2 minutes. Okay? So that is how important respiration is to any biological system. All right, okay, so allow me to share, I'll be using the same slides that you probably have seen before. Okay. Okay, can I confirm that you can see my PowerPoint slides?
Yes, Prof.
Okay, thank you, and they are moving? All right, okay. All right, so I was informed that you have gone through the lectures, and this will be a kind of a revision discussion. Okay, so I'll move through the thing very quickly. Okay, and if you have any question to ask or anything you would like to comment or discuss, feel free to raise your hand. Okay, and speak on the mic on the phone. Okay. So in this topic, we will cover 3 subtopics. Okay, which is the structure, importance, and formation of ATP. So respiration is all about the formation of ATP. It is a very important molecule, which carries energy in our living system. Okay. So just like in your cell phone uses a battery to carry energy and to function. Our body carries energy in the form of ATP. Okay, and use the ATP provided in a chemical form in the molecule of the ATP. So we will take a good look at this molecule okay, and see why is it so important. Okay. And in order to understand how ATP is synthesized in the body or in the cell, okay, we need to take a look at the redox reaction, okay, reduction oxidation reaction, okay, and finally we will dive into all the pathway and all the reaction that leads to the synthesis of ATP from glucose. Okay, so we have glycolysis, we have the link reaction, the Krebs cycle, the electron transport chain, and oxidative phosphorylation. I, I'm quite sure that this term are familiar to you all now. All right, and the learning outcomes. Okay, so these are important because this is what we expect of you all. That at the end of this lecture, okay, that you are able to state the different types how ATP is produced. Okay, basically there are two pathways: substrate level phosphorylation and oxidative phosphorylation. Okay, and you can explain, okay, oxidation and reduction reaction if you are asked a question, and you can describe, okay, how glucose is oxidized and the pathway by which glucose is oxidized, okay, glycolysis, pyruvate oxidation, and the citric acid cycle. Okay, I think the previous slide uses a Krebs cycle. Okay, and now we have, sorry, citric acid cycle. They are basically the same, different name. And finally, you should be able to describe the electron transport chain and chemiosmosis. So you find that at the end of the lecture, you are unable to fulfill this outcome, then you need to revisit the lecture and revise a textbook. Okay, so as a quick overview, so we start the discussion by talking about ATP, and then by revising redox reaction, and then we look at the stages of cellular respiration. Okay, and as we have seen in the previous slide, so we will go through glycolysis, okay, formation of acetyl-CoA from pyruvate, and then how acetyl-CoA is oxidized in the citric acid cycle, and finally how the electron extracted during these processes are used in the final step of oxidative phosphorylation to synthesize the bulk of ATP in the cell. And oxidative phosphorylation involves two steps, the first of which is the electron transport chain, followed by something called chemiosmosis. Are we okay so far? Am I going too fast or too slow? Anybody has any comment to say? Hello?
Okay, Prof.
Okay, thank you. All right. Okay, so we'll begin by looking at ATP. Okay, so you can see the molecular structure of the molecule on the small picture on top of the title. Okay, so ATP or adenosine triphosphate is the molecule that is used as the main source of energy for our metabolic function. So we are basically chemical beings. So our body function, yeah, due to the processes, biochemical processes that goes on in our cell. Okay, so and that those processes, respiration, okay, moving your muscle, okay, even thinking, as your brain is working, you may think that you're not using any energy as you sit down there and look at the slides that's there, or you might even be closing your eyes and sleeping. Okay, but as long as there's some activity in your body, as long as even your brain cells are thinking, you are consuming energy. And that energy is provided, okay, by using this molecule called ATP. Specifically, the energy is captured in the third phosphate bond. So if we look at the molecule, so you have an adenine side chain, okay, attached to a ribose sugar, and with 3-phosphate attached. So the third, the bond joining the second phosphate and the third phosphate is energy rich and can be fairly easily broken, okay, to release energy that can be used by other processes in the cell. So you can think of the ATP as tiny little batteries in the cell that can be shuttled from one part of the cell to another part, okay, and the energy can be used to perform different types of function. Just like you will use a battery in your torchlight or in your cell phone, okay, or in your game box or whatever, okay, to carry out different activities, okay, that you perform. Okay, so so as we mentioned a moment ago, so the energy stored in ATP is captured in the third chemical bond. Okay. So when this chemical bond is broken and releasing the phosphate, okay, the reaction is exogenic, meaning that it produces energy, and a total of about 7.3 kilocalories of energy per mole of ATP is released, okay, when ATP is hydrolyzed, okay, into ADP, adenosine diphosphate. So one phosphate is removed, okay, and the names change from triphosphate to diphosphate. The reaction requires addition of water, and that's why it's called a hydrolysis. So in this form, okay, this is how the cell uses the energy stored in the ATP molecule. Okay, so what we are going to discuss a lot today is how ATP is synthesized. Before you can use it, you have to make the ATP. Okay. So, and the way this body or, the cell, or our cell make ATP, okay, is to use energy that is released from catabolism, energy from breaking down larger molecules, okay, principally carbohydrates, which are energy rich. Okay, and when you break down those large molecules, energy is released, and that energy is captured in ATP, okay, by joining a phosphate, an inorganic phosphate group, the ADP, and capturing the energy in the third bond of the phosphate. And that ATP can later be sent to other parts of the cell, okay, and the bond can be broken again, and to release the energy that was stored, and that energy can be used to drive other cellular processes and metabolic processes. So the cycle is important that the cycle is always moving, okay, so that energy is produced in the form of ATP and used to drive cellular reaction in the cell, and that allows your body to function. So catabolic pathway that leads to the production of ATP. Okay, the main one will be cellular respiration. Okay. So when you're in your primary school, okay, you're probably told that everything requires oxygen. All living things require oxygen to survive, okay, to create energy. Then maybe further on in the secondary school, okay, you may be told that okay, some organism doesn't require oxygen. They can do fermentation. Okay, they can use fermentation to produce energy. And as you move on, okay, now we will tell you that there are organisms who doesn't require oxygen at all. So they can do what is called anaerobic respiration. Not only that, they don't require oxygen. In fact, oxygen is toxic to such organisms. Okay, but for the purpose of this topic, we will focus mainly on aerobic respiration, where oxygen is required to drive the synthesis of ATP. ATP is also produced in anaerobic reaction, okay, but the end receiver for the electrons is not oxygen, but another chemical compound. Okay, and in cellular respiration, the main substrate by which the cell break down to produce ATP is the sugar glucose. Okay. So it is mainly gained from the food that you eat, okay, in the form of carbohydrate or in the form of other sugars. Because every time you consume a sugar drink, a teh tarik, you are assimilating glucose into your system, and that glucose is then sent to the mitochondria to create ATP. So glucose is fairly important. It's very important for your well-being, because this is the main source of energy. Okay? But too much glucose is not so good. Okay? We'll see why in a moment. Okay, and this is basically the basic equation, okay, by which glucose is broken down into carbon dioxide and water, okay, with the requirement for oxygen molecule. Okay. And in the process, energy is released. So part of the energy is captured in ATP, and part of the energy is lost as heat. So the process of synthesizing ATP is fairly efficient, but not 100% efficient. And the part of the energy that is captured, okay, is lost as heat. And that's the reason why when you are very active, running around or doing some exercise, you sweat a lot. As the heat that you produce increases the body temperature. Okay, so besides carbohydrate, okay, which is provide the sugar, and which are then usually converted to glucose or other form of sugars that can be fed into the first part of the process called glycolysis. Okay, so the first part of cellular respiration is the process called glycolysis, whereby glucose is transformed into another substrate called pyruvate. Okay. So, the entry point for this process is the molecule glucose. But other sugars, okay, can be either isomerized into glucose and used directly, or they can enter into the pathway at different points. So sugar is the main substrate. Okay, and further on, pyruvate is converted into acetyl CoA, which is then fed into the citric acid cycle. So depending on which textbook you read, there are a number of names for this cycle. So the older textbook will refer to this as a Krebs cycle. So we have seen the names in one of the slides before this. Okay, and very old textbook may refer to this as the tricarboxylic cycle, the TCA cycle. I think during my time, it was called the Krebs cycle. It was about 30 years ago. Yeah. And then subsequently, it's called TCA, and now they call it the citric acid cycle. So it's the same cycle, just different name. Okay. And in the citric acid cycle, so a lot of what we call electron acceptors, okay, are reduced. Okay. So principally the molecule called NAD, which is reduced to NADH. Okay, and the electron that is captured in this molecule and a couple of other molecules is then used in the process of oxidative phosphorylation to drive the formation of ATP from ADP. So besides carbohydrate, okay, other food compounds can also be used for respiration. Okay. So fats can be converted into glycerol. So fats are basically triglyceride molecule, which you have a glycerol molecule with 3 fatty acids. So they can be split up into glycerol, and glycerol can enter into the glycolysis pathway directly as glycerol aldehyde phosphate. Okay, whereas fatty acids are beta oxidized and converted into acetyl CoA and fed into the citric acid cycle. So if your cell is starved for sugar, okay, for example, you are involved in some very intensive exercise, or you have not been eating well for the past few days and you're low on sugar, okay, then your body will start burning fat. That is why going to the gym and working out is a good way to reduce the fat. As you exercise and respire, fat is being burned, okay, to provide ATP. Okay. And when there's no sugar and not enough fat, then the body can actually use protein as an energy source. Okay. So proteins can be broken down into amino acids. Okay, and depending on type of amino acid, they can enter into the pathway, okay, at different points of the cycles. So certain proteins can be converted directly into pyruvate and enter at this point. Some proteins can be converted into acetyl CoA, and some amino acids can be directly fed into the citric acid cycle and take part in the oxidative phosphorylation process. Okay, am I good? Are we okay so far? Is there any question? Any comments?
No, sir.
Feel free to comment, okay, or feel free to share something if you have something that you think is interesting to share. Okay, all right. Okay, so during the processes, okay, ATP is produced by two pathways. Okay, so the first pathway is substrate level phosphorylation. Okay, so it's so named because the production of ATP by substrate level phosphorylation require a substrate with a phosphate group. Okay, so and the reaction is catalyzed by an enzyme. Okay, so whereby a substrate with a high energy phosphate, okay, is brought together in an enzyme, and the phosphate group is transferred to the ATP molecule to form ATP. So that's why we call it substrate level phosphorylation, because the substrate is involved. Okay, so and the transfer of a phosphate group, okay, from organic substance to ATP, and process is catalyzed by an enzyme. So a small amount of ATP is produced in this way, okay, both in glycolysis and in the citric acid cycle. So the main bulk of ATP is produced in the next process called oxidative phosphorylation. Okay, in order to understand oxidative phosphorylation, we need to do some revision on redox reaction. Okay, and there's a nice picture up here, a bit small. Okay, but it described oxidation and reduction well. Okay, so oxidation is where a compound, okay, loses electron, okay, and is oxidized. Okay. So oxidation involves the loss of electron. Okay, so the electron is transferred to another compound. Okay, and the compound that received the electrons is reduced. Okay. So redox reaction, okay, principally involve the transfer of electron from one compound to another. The compound that loses the electron is oxidized, and the compound that gains the electrons is reduced. Okay. So almost all the biochemical pathway in your body, okay, works based on the redox oxidation process. The energy is transferred from one compound to another, okay, and used in different part of your metabolism. Okay. So we have seen ATP can be produced by substrate level phosphorylation, where a phosphate is transferred from a substrate to ADP and to form ATP. A small number of the ATP are produced in such way. Most of the ATP in the cell is produced through the second pathway, oxidative phosphorylation, which involve a lot of a series of redox reaction, okay, and a series of transfers of electron between two reactants. So in the beginning, glucose is oxidized. Okay, so electron is taken away from glucose. Okay, and because of that, glucose is referred to as the reducing agent, because whatever that reacts with glucose becomes reduced. Okay, and at the end of it, all the electrons from that we take from glucose, okay, is transferred to oxygen. Okay, and in the process, oxygen is reduced because oxygen receives the electron. Okay, so oxygen is then called the oxidizing reagent. Okay, because whatever that oxygen becomes oxidized. Okay. So glucose, okay, there's a lot of things happen in between, but in general, glucose reacts with oxygen. Okay, so glucose becomes oxidized, electrons are transferred to oxygen, and oxygen becomes reduced. Okay. So you can think of it in a way that you can imagine that, although not 100% accurate, that a glucose contains a number of high energy electrons. Okay, and what respiration does is that it extracts all the electron and pass it to glucose. Okay, and along the way, the energy in the electrons is extracted in a very controlled manner to produce ATP. Okay, that's one quick question. So if we agree that glucose contains a number of high energy electrons, where does the energy come from? We are going to use glucose to synthesize ATP. We're going to transfer the energy from glucose to ATP. Okay, so glucose has a lot of energy. Where does energy come from in the beginning? Anybody want to try? Anybody? If the question comes in your exam. Where does energy to make glucose come from? That would probably been covered during your Form 5 biology. Okay, think about it. Okay, so glucose has a lot of energy. Okay, and we extract the energy by transferring the electrons. Okay, from glucose to oxygen. Okay, and as we mentioned, okay, it doesn't happen directly like this. Okay, so in between there's a lot of steps. Okay, and electrons are carried from glucose to the oxygen, okay, through a number of electron carrier, principally something called NAD plus that we'll talk about in a short moment. Oh, okay, thank you. There's an answer, sunlight. Yeah, that's correct. Okay, so the energy to make glucose comes from sunlight during photosynthesis. That's where the energy is captured. Okay, and we now extract the energy and use it to make ATP. So you can say that most of the life form on Earth depends on the sun for energy. In fact, during my time, we say that every life form on Earth depends on the sun for energy. But then later on we find that that is not 100% true. There are a few organisms that we know that doesn't depends on the sun for energy. But that's not a story. Okay, so this is NAD plus. This is the one that will carry the electron from glucose all the way to oxygen. Okay, so NAD plus is nicotinamide adenine dinucleotide. Okay, it's one of the major electron carrier in the cell. There are a few more, NADP, FAD, and so on. Okay, so NAD is technically an electron acceptor and an electron carrier. So it can accept electrons from a number of compounds, okay, and carry the electrons to another pathway where they are assimilated by other compounds. Okay, so NAD plus function as an oxidizing agent because it removes electrons, okay, and it's also a common coenzyme, okay, meaning that it helps other enzymes to perform their function. Okay, so how does NAD plus function? Okay, so you can imagine that, okay, NAD plus is a receptor for electron. So when something needs to be oxidized, okay, so enzymes called dehydrogenase. Okay, so from the name you can guess that, okay, it's an enzyme that remove hydrogen, okay, hydrogenases, okay, dehydrogenases, okay. So these enzymes, what they do is they remove a pair of hydrogen atoms. And if you look back in the chemistry lectures, you know that a hydrogen atom consists of one electron and one proton. Okay. So one electron circling a nucleus of one proton. Okay. So if a pair of hydrogen atoms is removed, then we are removing two electrons and two protons. Okay. So diagrammatically, it's shown as two electrons with a negative charge and two protons with a positive charge. Okay. A hydrogen ion is a proton. A very basic particle. Okay, so this is taken from the substrate, and two electrons and one proton is passed to NAD plus. Okay, so NAD plus receives two electron and one proton and become reduced to NADH. Okay. The extra proton, okay, is not taken up and diffused into the surrounding matrix. Okay, so from NAD plus, okay, after receiving two electrons and one proton, it becomes NAD. This is a reduced form of NAD plus. Okay, so the electron is now captured by NADH. Okay, and the energy is stored in NADH for other purposes. Okay, so that basically the redox reaction that happens a lot, okay, during the process of cellular respiration. Okay, so NAD is formed in almost every step of the process of cellular respiration. Okay, in glycolysis, the first stage, where glucose is converted to pyruvate. This happens in the cytosol or the cytoplasm of the cell. Okay. And after pyruvate is formed, okay, it's then transported into the mitochondria. Okay, and in the mitochondria, pyruvate is changed into acetyl-CoA. Okay, in a reaction that we call the link reaction or pyruvate oxidation. And acetyl-CoA is then fed into the citric acid cycle. Okay. And this is where the major portion of NADH and also another electron carrier, FAD plus, is reduced to FADH2 is produced. Okay. This happens in the mitochondria. Okay. And the last step, okay, all the electrons that's captured by NADH and FADH2 is then used in oxidative phosphorylation for ATP synthesis. Okay, in the two-step process, okay, involving electron transport, chain, and chemiosmosis. And this again happens in the mitochondria, by different part of the mitochondria. Okay. So this is a quick synopsis of what happened, okay, to synthesize ATP in the mitochondria. Okay, so let's look at it in the form of a diagram. Okay, so the first process, of cellular respiration begin with glucose, which is converted to pyruvate in the process called glycolysis. So this process produces ATP, a small amount of ATP through substrate level phosphorylation. It also produces NADH. All right, and then in the next stage, pyruvate is transported into the mitochondria and converted into acetyl-CoA. I think NADH is also produced here, but probably it's not written here. Okay, and then acetyl-CoA is shuttled into the citric acid cycle, and this is where most of the NADH and FADH2 is produced, and also a small amount of ATP is produced through substrate level phosphorylation. And in the final step, okay, all the NADH and FADH that is produced from the glucose is then taken to the electron transport chain, okay, where they are used to produce most of the ATP through oxidative phosphorylation. So we will look at this all four processes, okay, in detail in a short while. Okay, but just to recap, okay, ATP can be produced through substrate level phosphorylation, which happens during glycolysis and also during the citric acid cycle. Okay, but the bulk of ATP is produced using oxidative phosphorylation, okay, which happens during the electron transport chain and chemiosmosis, during when the electron that is captured from the different intermediate molecules of glucose is used to drive the process of ATP synthesis. Okay, so let's begin by looking at glycolysis. Okay, before I carry on, any question?
Prof, we have one question from student.
Okay, sorry, I missed
Okay.
that. Okay.
All right. So, first question from Aizat. What is the connection between phosphorylation and substrate level phosphorylation?
Okay, it's just it's meta terminology. So phosphorylation is the term that is usually used to describe the attaching of a phosphate group to a molecule. So when you attach the third phosphate group to ADP, adenosine diphosphate, that is the process of phosphorylation. Substrate level phosphorylation is the term that we use to describe the synthesis of ATP by transferring a phosphate group from a substrate to ADP. So I hope that answered the question. Okay. All right. Thank you. All right. So, okay, I'll move on. Okay. So we'll begin by looking at the first stage of respiration. That is the process of glycolysis, where glucose is broken down into pyruvate. Okay. So glycolysis literally means sugar splitting. Glyco is sugar. Lysis is breaking up something. Okay. And that describes the process of breaking down sugar into two molecules of pyruvate. Okay. And this process happens in the cytoplasm, okay, or cytosol. Okay, and it involves two major phases. The first phase is called the energy investment phase, okay, where you need to use ATP to split glucose into two. Okay, and this is followed by the second phase, which is called the energy capture phase, okay, or the energy payoff phase, where after splitting the glucose, you can now use the two intermediates molecule to produce 4 ATP. Okay. So you have a net gain of 2 ATP here. Okay. So glycolysis occurs whether or not oxygen is present. So the process does not require oxygen, okay, and it can happen whether there is oxygen or not in the cell. So the small picture here shows where how glucose, a 6-carbon atom, is broken down into 2 molecules of pyruvate, which are 3-carbon sugars. Okay, and the diagram show how 2 ATP is used in the first stage, okay, to break glucose into 2 intermediates, and during the subsequent pathway, okay, so 4 ATP is formed. Okay. We will look at the detail later on how this happened. Okay, and also notice that besides ATP, okay, 2 NADP is 2 NADH is also formed. So the net yield from glycolysis, okay, is that one molecule of glucose is converted into 2 molecules of pyruvate. Okay. And in the process, 4 ATP is produced. Okay, and remember 2 ATPs is used before you can get your 4 ATP. So you have a net gain of 2 ATP. Okay. And also 2 NADH is produced during the process. So okay, I won't go into so much into the detail. Okay, you want to know whether you need to memorize all these details. Okay, you can ask your lecturers. Okay, I would say that during my days, okay, 30 years ago, we are required to memorize all of this. Okay, right down to how many carbons are there in the intermediate. Okay, but I think you are of a different generation, so you may not be required to memorize. Ask your lecturer. Okay, so we'll just very quickly run through the process where glucose is in the first step converted into glucose 6-phosphate, and that requires 1 ATP. Okay, and then glucose 6-phosphate is then isomerized into fructose 6-phosphate, and then another ATP is added, and you have fructose 1,6,2 bisphosphate. Okay, so notice that what this does is to attach 2 phosphate group to the glucose molecule, okay. And these are the phosphate group that will be used for the synthesis of ATP later on. Okay. And at the last step, okay, fructose biphosphate is splitted in, from a 6-carbon molecule into 2 3-carbon molecule. Glyceraldehyde 3-phosphate, okay, and dihydroacetone phosphate, which is later isomerized into glyceraldehyde 3-phosphate. So the net outcome of this part of the process is the production of 2 molecules of G3P, each carrying a phosphate group. Okay. And in the next step, okay, so NAD oxidized G3P into 1,3-biphosphate glycerate. Yeah. Again, 2 more molecules of phosphate is added to the each of the G3P. Okay, and now you have a 3-carbon structure, each with 2 phosphate. So altogether 4 phosphate groups are added. Okay, and this is then later used to synthesize 4 ATP molecule. Okay, and finally producing pyruvate. So here ATP is synthesized using substrate level phosphorylation, okay, by transferring the phosphate group from the intermediate substrate to ADP, okay, and capturing the energy in the 3rd phosphate bond. Okay. So the net outcome, okay, is, as you mentioned, 4 ATP minus the 2 ATP that you used in the beginning, okay, and 2 NADH. Okay, so that is the a quick look at glycolysis. Right, next, once pyruvate is formed, okay, then it is shuttled into the mitochondria, okay, and this is where the bulk of ATP synthesis happen. Okay. So pyruvate is transported into the mitochondria, okay, and undergoes oxidation. Okay, so this is also called a link reaction. Okay, and from pyruvate, okay, acetyl-CoA is produced, okay, by an enzyme called pyruvate dehydrogenase, and acetyl-CoA is then transported to be used in the citric acid cycle. Okay, so this is the process where pyruvate, okay, here 1 carbon is removed, okay, and becomes carbon dioxide, okay, and the 2-carbon acetyl group is then coupled with a coenzyme A to form acetyl-CoA, and in this process 1 NADH is produced. Okay, so the next step is where the bulk of the electron acceptors are reduced, okay, and it is called the citric acid cycle, and as we mentioned a moment ago, okay, it's also called the Krebs cycle or the tricarboxylic acid cycle. So different names but same cycle, and this happens in the mitochondria matrix. So this is a picture of Hans Adolf Krebs. He is the one that discovered and also found out most of the reaction that is involved in the Krebs cycle. Okay. So and the process happens in the matrix of the mitochondria. So this is a cutaway diagram of the mitochondria, an organelle in a cell. Okay, so it consists of 2 layers of membrane, an outer membrane and an inner membrane. Okay, and the brown color part here is called the intermembrane domain. Okay, region. Okay, where a lot of reaction happens. Okay, you'll see in a moment. All right, and the blue part is the internal part of the mitochondria, which is called the matrix. So the citric acid cycle happens in the matrix. Okay. But the next step of the reaction, the electron transport chain and chemiosmosis happens on the inner membrane. Okay, and this is how the citric acid cycle looks like. Okay, so there are 8 steps involving 8 intermediates. Okay, so acetyl-CoA enter, okay, and it's picked up by oxaloacetate, okay, and then it's transformed to citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and finally you recycle back as oxaloacetate, ready for the next acetyl-CoA. Okay. So that's why it's called a cycle. Okay. As long as there is a supply of acetyl-CoA, the cycle will continue. So again, we will not go into much detail on the biochemical part. Okay, I believe that you can read that up. Okay, so what we are interested in is what is produced during this cycle. Okay, and we can see that at 3 parts of the process. Okay, NADH is produced. So each round of the cycle will produce 3 NADH. Okay, 1 ATP is produced by substrate level phosphorylation. And also 1 FADH2 is produced. FAD stands for flavin adenine dinucleotide. Okay. So it's another electron carrier similar to NAD, okay, but it accepts 2 hydrogen ions and form FADH2. Okay, so the net outcome of the citric acid cycle is 1 ATP, 3 NADH, and 1 FADH2. Okay, so if you're interested in the details, you can read the next few slides. Okay, so how the carbons are assimilated into the cycle, get transformed, okay, to other intermediates, and in the process, okay, NAD is reduced to NADH. Okay, so remember that every time NAD is reduced to NADH, okay, 2 electrons is captured. Okay, so in the process, okay, carbon dioxide is also produced. So some carbon is lost from the backbone of the intermediate compounds. Okay, and at step 5, 1 molecule of ATP is produced by substrate level phosphorylation. Okay, so during the conversion of succinyl-CoA to succinate, okay, 1 phosphate group is joined to another molecule called GDP, guanine diphosphate, which look very similar to adenosine diphosphate. It's just that the adenine group is replaced by the guanine group. Okay, and by addition of 1 phosphate group, okay, guanine diphosphate becomes guanine triphosphate. Okay. So the energy is stored in the 3rd phosphate bond, just like in ATP, and then that phosphate group is transferred to an ATP to form ATP. And that's 1 molecule of ATP formed during the citric acid cycle. Okay. And at the same time, as step number 6, 1 molecule of FAD is reduced to FADH2. So again, 2 electrons are picked up here. Okay. And finally, another NADH is produced at step 8. Okay. So just to quickly recap, okay. So throughout the process, during the process of converting pyruvate to acetyl-CoA, the link reaction or the pyruvate oxidation reaction, 1 NADH is produced. Okay. And when acetyl-CoA enters into the citric acid cycle, 3 NADH, 1 FADH2, and 1 ATP is produced. So a total of, okay, from between the mitochondria, okay, from the process of pyruvate to acetyl-CoA and citric acid cycle, you have a total of 8 NADH, 2 FADH2, and 2 ATP being produced for each molecule of glucose oxidized. And not to forget, so that during glycolysis, 2 NADH is produced, and also 2 ATP is produced. Still quite a small number of ATP so far. Okay, but remember that most of the energy is still captured between the electrons that is now being carried in the NADH and 2-FADH2. Okay, so in the last part of the process, okay, are we okay? Am I going too fast? We seem to be
Okay,
covering.
Prof.
We seem to be covering things quite fast. Should we stop for a break?
Okay.
Okay, all right. So
Right.
should we stop for a 10-minute break, and then we come back and tackle the last part of the cycle?
All right.
Okay,
So,
thank you.
okay, student, we will have 10-minute breaks, and our lecture will continue at 11.
Okay, thank you. See you again.
Okay, hello everyone and welcome back. So, okay, Prof, shall we continue?
Okay, thank you, Dr. Manzani. Okay, so welcome back. Okay, and we will pick up from where we left off just now. So in the previous hour, we wanted to make ATP. So we took a glucose molecule, okay, and we broke them into two molecules of pyruvate. Okay, in the process, we managed to get two ATP. Okay, and then we converted the ATP, the pyruvate into acetyl CoA, and sent it into the citric acid cycle. And in the process, we get another two ATP. So far, altogether, we have four ATP. Yeah, after working so much with glucose. Okay. And if I'm a cell, I won't be happy, okay, because 2 liter ATP. Yeah, I won't be able to survive on that. Okay. I definitely need more ATP. Okay, but then fear not. Okay, most of the energy from glucose has actually been transferred into the NADH and the FADH2. So the energy is captured in the electrons that has been received by these two molecules. And in the next process, okay, we will see how, okay, we use the electron transport chain to extract the energy from this electron. And then in the last step in chemiomosis, we will see how that energy is then used to synthesize the bulk of ATP, okay, that the cell will obtain from the mitochondria. So you have to imagine that this happens in every single mitochondria in your body. Okay? And you have about 1 billion cells. Some cells may contain a few mitochondria. Some cells can contain up to 50 and 100 mitochondria. So at any moment, okay, about 50 billions of such reaction is happening in your body. Okay? So that's the kind of numbers that of ATP that you're producing, and the numbers of glucose that you're breaking down, okay, every moment, in order for you to move around and do activities. Okay, so in the next stage, okay, we'll talk about oxidative phosphorylation, the second pathway that is used to make ATP, and this involves the electron transport chain, okay, and chemiosmosis, two parts of the process. There's a nice animated video here. We'll come back to this later on. Okay. All right. So when I first saw this process 30 years ago, I was totally awestruck. To me, it's a very intriguing process. It's something so fine-tuned, okay, and can be happening at a cellular level, okay, unguided. Okay, they just move on their own, okay, or automatically, okay, and they seem to know, okay, what to do. Okay, so the first part of the oxidative phosphorylation involves something that is called the electron transport chain. Okay, and the electron transport chain is performed by a series of about 6 proteins. Okay, so we have complex 1, 2, 3, 4, four complex of four multi-protein complexes. Okay, so each complex contains a number of proteins working together, okay, in order to achieve a certain function. Okay, we also have complex 2. Complex 2 has been mentioned. We also have coenzyme Q, which is electron carrier, a mobile carrier, in the sense that it can move around. It can move, while the other proteins are embedded in the inner membrane. Coenzyme Q and another protein, cytochrome C, can move between these proteins and transferring electrons from one protein to the other. Okay, so that forms the physical part of the electron transport chain. This happened at the inner mitochondrial membrane. Okay, so the yellow part down here is the matrix of the mitochondria, and the slightly dark orange part here is the intermembrane space. Okay, and somewhere up here you will have the outer membrane. Okay, so the complex 1, 3, and 4 are embedded in the inner membrane, and that is actually essential for its function. But we'll come back to the chemiomosis later on. Okay, so we just focus on the electron transport chain at the moment. Okay, so the electron transport chain is a collection of protein complexes and organic molecule embedded in the inner membrane. Okay, or sometimes when it's folded into the matrix, it's called a cristae. The structure that where the inner membrane projects into the matrix, okay, is called a cristae. Okay, and you can find all the proteins that complexes involved in the electron transport chain get attached to the inner membrane. Okay, and this is a blow up diagram of what actually happens. Okay, so you can see that we have NADH here, we have FADH2 here. So these are the electron carriers that has captured the electron as we break down glucose step by step all the way through the citric acid cycle. So NADH will pass its electron, okay, to complex 1. Okay, so it enters the ETC, the electron transport chain, through complex 1. Whereas FADH2 pass the electron through complex 2. Okay, so there are two different entry points into the electron transport chain. Okay, NADH enters through complex 1 and FADH enters to complex 2. So from complex 1, the electron is passed to coenzyme Q, or the old name is ubiquinone. Okay. So this is the reason why if you have been approached by MLM agents trying to sell you coenzyme Q as a supplement to boost your energy, okay, there is some scientific truth to it, because coenzyme Q is involved in the electron transport chain that is involved in generation of energy. But how far does the coenzyme Q that you eat actually reach this point? That is the question that we cannot answer yet. Okay? So you can consume the coenzyme Q. It may sort of help you to boost energy by increasing the level of your coenzyme Q. Okay, and coenzyme Q function to pick up the electron from complex 1 and 2 and then pass it to complex 3. Okay, and then the electron is passed to cytochrome C, which shuttle it to complex 4. And finally, the electron is picked up at the end by oxygen. Okay, so the electron has to go somewhere. A free electron is something that's dangerous to the cell, but they are very reactive, and they can damage a lot of components of the cell if they are not properly dealt with. So the electrons that's released from the NADH or FADH2 will finally be picked up by oxygen. Okay. That's why oxygen is required in the process. Without oxygen, okay, the process will not happen. Okay, so oxygen sort of pull the whole reaction. Okay, so in the absence of oxygen, okay, this process do not happen. Okay, all right. So that is basically what happened during the electron transport chain. Okay, and in complex 1, 3, and 4, okay, as the electron move through the complex, okay, so the energy is released in a very controlled manner. So bit by bit, the energy is released in a controlled manner, and that energy is used by complex 1, 3, and 4 to pump one hydrogen ion from inside the mitochondria out through the inner membrane into the intermembrane space. Okay, so all this is just for one purpose, okay, to allow the electron transport chain to pump 3 hydrogen ion from inside the matrix into the intermembrane space. Okay. So seems to be a lot of things to do just for 3 simple results. Okay, pumping of 3 hydrogen ion into the intermembrane space. Okay. All right, and this is a much more detailed look at the electron transport chain. So you will see that between complex 1, okay, and 2 and 3 and 4, there are a number of other electron transport agents, okay, electron carriers. Okay, so you have a flaming mononucleotide in complex 1. You have an iron sulfur protein in complex 1. Okay, okay, then you have a coenzyme Q, which carries the electron from complex 1 and 2 to 3. And in complex 3, you have cytochrome B, another iron sulfur protein, and cytochrome C1. And then the electron is passed to cytochrome C, okay, and passed to cytochrome A and A3 in complex 4 before it's finally passed to oxygen. Okay, so oxygen in this case is the most electronegative component here. Okay, so the electron will tend to move towards oxygen. Okay, as this is the most electronegative. NADH and FADH are the least electronegative, okay, compounds here. Okay, and the electron will tend to leave NADH and move towards oxygen. Okay, but notice that the entire process is very much controlled. Okay, At no point is the electrons allowed to roam free. They are always passed from one carrier to another carrier, okay, in a very controlled manner. And during the passage of the electron, as it's being transported down the electron change, okay, energy is released, okay, in bit by bit, okay, part by part, yeah, and that is used to pump out in a hydrogen ion, okay, from the matrix into the intermembrane space. Okay. So, and each of these electron carrier will undergo the same process. They will be oxidized, reduced, okay, as they donate or accept electrons. Okay. So what happened what is the net gain of this process? Why do we spend so much effort and energy just to pump the hydrogen ion out? Okay. So just to recap, so in the first stage, NADH and FADH donate electron to the electron transport chain. Okay. Right. And the electron are passed from one electron carrier to another through a series of redox reaction. Okay. And in the process, energy is released. Okay. Before the electron finally is picked up by oxygen to form water. Okay. The final electron acceptor is oxygen. Okay. And during the process, energy that's released is used to pump a proton, a hydrogen ion, from the matrix into the intermembrane space. Okay. And then because of that, where the electrons, the energy-rich electron from NADH and FADH2 gradually lose the energy as they go along. Okay. So we are in fact extracting the energy from the electron, okay, slowly, okay, part by part, bit by bit. Okay. And the net result is that we set up a gradient of proton across the inner membrane. So if we keep pumping up protons, hydrogen ion, okay, we will create a situation where, okay, in the intermembrane space, there will be a high concentration of proton. And in the matrix, there will be a low concentration of protons. And that sets up what we call an electrochemical gradient. Okay. So all this is just to set up this electrochemical gradient. So you have a high concentration of proton on the outside and a low concentration of proton on the inside. And when you have such a situation, what happens? You are creating an electrochemical gradient and you are storing potential energy in that gradient. So that potential energy is stored in the proton. And in the next step, this potential energy will be converted to kinetic energy, and that is used to synthesize ATP through the process of oxidative phosphorylation. Okay, so the last step is chemiosmosis, okay, where the energy is coupled to a molecule and used for the production of ATP. Okay. So chemiosmosis is a process of producing ATP using energy in the proton electrochemical gradient. So you have a high concentration of proton in the intermembrane space, okay, and this proton gradient, okay, sometimes also called the proton motive force, okay, has potential energy, okay, and this energy is then used to drive the ATP synthase, okay, another protein complex in the inner mitochondrial membrane, okay, to synthesize ATP from ADP and phosphate. Okay. So what actually happened is that due to the gradient, okay, so protons from the intermembrane space will have a tendency to flow into the matrix, okay, and this is allowed to happen through a small channel in the ATP synthase. Okay. So, and when that happens, okay, so that energy from the moving proton, okay, is used to drive a molecular rotor, okay, in the ATP synthase. So it's actually something that rotates. If you look at the molecule modeling, okay, it's shaped like rotor, and it can rotate when the proton move past it. Okay, so the proton will diffuse back into the matrix, okay, and cause the synthesis of ATP from ADP and inorganic phosphate. Okay, so this is just a larger diagram of it. So electron is transport and pumping a proton create a proton gradient across the membrane. Okay, and ATP synthesis is powered by the flow of proton back across the membrane. Okay. So first you set up a gradient, and then you use the energy stored in the gradient to synthesize ATP. It's as though you are pulling a rubber band, okay, and that energy is stored in the rubber band. And when you release the rubber band, that energy can be used to synthesize ATP. Okay, and this is how the ATP synthase look like at the molecular level. So it has three components. Okay, a stator, okay, which hook the molecule to the inner membrane and keep it in place. It has a rotor here, okay, whereby every time a proton moves from the intermembrane space into the matrix, it has to go through a certain channel only, and is picked up by the rotor, and the kinetic energy actually turns the rotor one full cycle before the proton is released and enters into the matrix through another channel. Okay, so you can imagine the proton as water, okay, entering into a rotor and spinning the rotor, okay, before it's released into the matrix. Okay, so you can even imagine that it looks something like the hydroelectric power station that we have, okay, in Cameron Highland and in Sarawak, where water at high level is used to spin the turbine, okay, and then the turbine is used to turn the dynamo to generate electricity. So the same thing, almost the same thing happens here, okay, but instead of a flow of water, a flow of proton is used to spin the rotor, okay, and that energy is then transferred to the catalytic knob, okay, and used to synthesize ATP from ADP and inorganic phosphate. Okay, so that's basically complete the whole story of how, okay, glucose is used to synthesize ATP. Okay, how the electron from glucose can be extracted and then taken through a whole series of processes, and finally used to create a proton gradient, and then the energy stored in the proton gradient is used to turn the ATP synthase and produce the bulk of ATP through a process of oxidative phosphorylation. Okay, so how many ATP in the end do we get? Okay, so we already know that we get 2 ATPs from glycolysis and 2 ATP from the citric acid cycle to substrate level phosphorylation. Okay, oxidative phosphorylation produce another 26 to 28 ATP. Okay, so you combine this all together, so per molecule of glucose, okay, you have about 30 to 32 ATP. Okay. Why 30 or 32? Okay, that depends on one step here. Okay, remember 2-NADH is produced during glycolysis, and this is also channeled to the oxidative phosphorylation reaction. Okay, so depending on how they enter the mitochondria, okay, that determines whether you can get 26 or 28 ATP from oxidative phosphorylation. Okay, so we'll take a look at that in a short while. Okay, so okay, all right. Okay, so each NADH generates proton motive force to synthesize 2.5 ATP. This is by calculation. We don't really have any good data to back it up, but we think that based on a chemostatic calculation, one NADH will provide enough energy to synthesize 2.5 ATP. During my time, it's 3 ATP, but I think in the past 30 years, they have refined the calculation, and now we have decided that, okay, 1 ATP equivalent to 1 NADH is equivalent to 2.5 ATP, and one FADH2 is equivalent to 1.5 ATP. Okay. All right. So the difference of 30 or 32, okay, depends on how the NADH produced through glycolysis. Remember, glycolysis happened in the cytosol. Okay, in order for these two NADH to take part in the oxidative phosphorylation process in the mitochondria, okay, it has been taken into the mitochondria using active transport, okay, and in some cells, okay, I think in the wait just a moment, in the liver cell, for example, okay, so the NADH is taken into the cell, okay, through a malate aspartate shuttle, I believe, okay, and the electron is transferred to NADH. Okay, so there's no energy loss here. Okay, so to NADH, okay, and you get to NADH, and this is then taken into the electron transport chain. Okay. However, in other cells, for example, in the brain cell, okay, the NADH is taken into the mitochondria using a different process. I think it's taken in using a glycerol phosphate shuttle, okay, and that process is not so efficient. So and the electron is picked up by 2-FADH2 instead of 2-NADH. Okay. So in the brain cell, okay, so instead of getting NADH, you get FADH, and this result in a slight loss of ATP because one FADH can only produce 1.5 ATP, whereas one NADH can produce 2.5 ATP. Okay. So depending on which process that is used to transport the NADH, okay, into the cell, okay, you end up with either 32 or 30 ATP in total from each molecule of glucose. So I think the calculation is given in more detail here. All right. So from glycolysis, we use 2 ATP, but we generate 4 ATP. So there's a net gain of 2 ATP from substrate level phosphorylation. Okay. From pyruvate, from glycolysis, we also get 2 NADH. Okay, and each NADH is equivalent to 2.5 ATP. Then we have 5 ATP here. All right. During the conversion of pyruvate to acetyl-CoA, you generate another 2 ATP, so another 5 ATP. Okay, and these are all channeled to the oxidative phosphorylative pathway. Okay, and when acetyl-CoA is taken into the citric acid cycle, you generate another 6 NADH and 2 FADH2, okay, which is equivalent to 15 ATP and 3 ATP. And also 2 ATP is produced by substrate level phosphorylation using GTP during the citric acid cycle. Okay, so a total yield of 32 ATP from one molecule of glucose. Okay. All right, and if the cell uses a different shuttle to bring in NADP from glycolysis, and which is converted to FADH, then you have a lower yield of 30 ATP. Okay. Okay, so this is what happened. Okay, this is why there can be a difference in the number of ATP per mole of glucose. Okay, it depends on what the cell used to transport the NADH from glycolysis, okay, into the mitochondria to take place to participate in the oxidative phosphorylation process. Okay. If it's taken in as NADH, okay, if the NADH from glycolysis pass the electron to NADH in the mitochondria, then you have no energy loss and you get 32 ATP. However, if the NADH from glycolysis transfer the electron to FAD in the cell, in the mitochondrion, then you have a lower ATP yield of 30 ATP per glucose molecule. Okay, and just a recap of the entire picture. So from glucose, through the process of glycolysis, you get 2 pyruvate, okay, and which is converted into 2-acetyl-CoA. Okay, so in glycolysis, you have 2 NADH being produced. Okay, so and a net gain of 2 ATP. Sorry, too eager to finish. Okay, and then during the conversion from pyruvate to acetyl-CoA, okay, you produce 1 NADH. Okay, and when acetyl-CoA enters the citric acid cycle, you produce 3 NADH, 1 FADH2, and 1 ATP, okay, per molecule of acetyl-CoA. And since one glucose produce 2 molecules acetyl-CoA, So you have a total of 6 NADH, 2 FADH2, and 2 ATP. And when these are all taken into the oxidative phosphorylation pathway, then you end up with a total of 32 ATP molecule for every molecule of glucose that is oxidized. And the electrons are finally passed to oxygen. Okay, so I think that completes the story of respiration, of how we break down glucose, okay, to generate ATP, okay, and how we gain 32 molecules of ATP from one molecule of glucose. Okay, in reality, it's not that perfect. So some energy is lost, and some of the process is not fully efficient. So you may actually get less ATP. We still don't have a good idea of how many ATP that we actually get from a molecule of glucose. By calculation, it's 32 ATP. But we have to take into account that every time something is transported in and out of mitochondria, okay, some ATP is used, some energy is used. So the actual yield, okay, could be less. Okay. So I think the scientists out there are still working out, trying to figure out, okay, what is the actual number of ATP that is produced when one molecule of glucose is oxidized. Okay. So besides producing ATP, okay, the citric acid cycle is also important in the sense that, okay, many of the intermediates compound that is produced in the citric acid cycle are important for other parts of the cell, for other cellular function. Okay, so a lot of them form backbones, okay, for the synthesis of other compounds like amino acids. Okay, so if a cell has enough ATP, okay, some of the compounds that is formed in the citric acid cycle, like the alpha ketoglutarate and so on, can be actually shunted, okay, to make other molecules that is required by the cell. Okay. All right, and is there anything else that we miss? Okay, so and we have seen how that the process requires oxygen. Okay, oxygen is extremely important to us, okay, for just one purpose, that they take up the electrons at the end of the reaction. Okay. So there seems to be a sort of an anticlimax that a molecule so important as oxygen that we absolutely cannot live without, okay, has only that one function, to pick up the electrons that is extracted from glucose. Okay. All right. Any question? So.
Thank you so much, Prof. We have several questions here. So first question, what maintain the citric acid cycle?
Sorry, what maintain the citric acid
Yes,
cycle?
the questions from Aizat. What maintain the citric acid cycle?
Okay, I suppose you mean what regulates the citric acid cycle.
I think so.
Okay, so the cell has a very strict, generally has a strict regulatory pathway. Yes, so when energy is required, okay, so the citric acid cycle will be used mainly for the synthesis of ATP. Okay. However, when energy is abundant, when there's enough ATP, yeah, being synthesized, there's a lot of glucose, there's a lot of oxygen, okay, then the citric acid cycle can be shunted for other purposes. Okay. So, if I'm not mistaken, for the cell, energy generation is always of priority. So in terms of, in the case of low energy, okay, the citric acid cycle will always be used, okay, for the generation of ATP first, okay, before other purposes. I hope that answered the question.
Okay, then second question, part of cellular respiration where oxygen is needed is during oxidative phosphorylation. Can link reaction and Krebs cycle occur without oxygen?
I think the general evidence now point to the fact that the only part of the cycle that can do without oxygen is glycolysis. So for the other reaction to happen, you need oxygen. So without oxygen to pull the electrons, okay, so the entire chain will not happen.
Okay, and third question, where is the ADP and inorganic phosphate come from?
Okay. Uh, uh, you can imagine that they're always in the cell, in mitochondria. Yeah, so they are synthesis by other pathway. Okay. And remember that in the reaction, they are not broken down or changed to other chemical during the metabolic reaction. So ATP is broken down into ADP and phosphate, and then they're recycled back into ATP. Okay? So there's always a pool of ATP and inorganic phosphate in the mitochondria. Okay? So where they are continuously broken down into ATP and phosphate and resynthesized into ATP.
Okay, and last question, Prof. How NADH and FADH2 produce ATP?
They don't produce ATP directly. So the production of ATP is through the oxidative phosphorylative pathway. Okay, so NADH and FADH2 are mainly electron carrier. So their job is to capture the electron when we break down glucose in the citric acid cycle, okay, and then bring the electrons through the electron transport chain. So the electron transport chain extract the energy from the electrons. So you can see that once the NADH has transferred the electron to the complex, okay, they become reoxidized to NAD plus, and they go back to the citric acid cycle to pick up further electrons. Okay. So the synthesis of ATP, okay, is through the uh, energy contained in the electron that is extracted by the electron transport chain and converted into the potential energy of the proton gradient, of the electrochemical gradient across the inner membrane. Okay. And that is later transformed into kinetic energy as a proton flows back into the matrix and spin the rotor, and that converts the energy that is used by the catalytic knob to synthesize ATP from ADP and PI and phosphate. So there's a lot of energy transformation. So chemical energy in the electron is converted into potential energy of the gradient, which is then converted into kinetic energy of the proton flow, and then finally converted back into chemical energy stored in the phosphate bond.
So, okay, thank you very much, Prof. So I think that's all that question that we have for today.
Okay,
Thank you,
in that
Prof.
case, can I ask a question just to pick your minds a bit? Okay, so we have seen that oxygen is important, but only as the final electron acceptor. Okay, so what do you think? Can we use something else besides oxygen? Since that's its only function, to pick up the electron. Anybody want to comment? So this is the role of oxygen at the very end of it, to pick up the electron. Simple function, but extremely important. So much so that we can't live for 2 minutes without oxygen. Okay, maybe 5 minutes. Okay. So can oxygen be replaced? Can we replace oxygen with something else? What do you think?
Can I answer?
Yes, please.
I don't think that the oxygen can be replaced, because if we replace the oxygen, then the end product of the cellular respiration will be something else and not water.
Okay, thank you. Okay, so that's true in human. So we can't replace oxygen. Okay. But there are other organisms that has successfully done that. So there are bacteria that can actually replace oxygen with something else. Okay. So we have bacteria that can use in place of oxygen a number of other compounds like sulfur, sulfide, nitrite, and so on. Okay, so these are very weird bacteria that instead of glucose they eat iron. Okay, and they transfer the electron through the almost the same process. Okay, to sulfur or nitrate and so on. Okay, so life has a lot of surprises. Okay. We as human dah lebih manja. Okay. No, nak oxygen sahaja. Okay. And also because oxygen is the most electronegative acceptor. So using oxygen, you get the most yield. You get 32 ATP. Using anything else, the process will probably stop somewhere in the middle. Okay, because uh, it doesn't have such a strong pull as oxygen. It's not as electronegative, and you have a lower number of ATP being produced. Okay, right. Thank you very much. Okay, very good. Okay, okay. Now, do I want to ask you another question? See if we have time. Okay, I think I will not bother you people anymore. Okay, so okay, Dr. Kaniza, I'm done.
Okay, thank you, Prof, for extensive explanation about cellular respiration. So I hope today, I hope I hope today's lecture is beneficial to all of us, and I'm sure we have learned so many things from Prof today. So again, thank you so much, Prof, for spending your time today with us, and I hope we can have you again in the future, inshallah. So to student, you can find attendance links in the chat box, and QR code will be displayed on the screen shortly. Thank you everyone for joining our lecture today. Stay safe and take care. Goodbye and Assalamualaikum.
Okay, thank you very much, everyone. Okay, I apologize if I started on certain point. Okay, so it's been a long time before I visit this topic, and I still find it very intriguing. Okay, so thank you again for inviting me for this lecture.